Bismillahirrahmanirrahim. We start the new chapter, chapter 16, and hopefully I should discuss 16.1 component with you all. And this is 5090 O-level GCSE Biology, new syllabus 2023. Now, as we look at this chapter, chapter 16 is development of organisms and continuity of life. The first is nuclear division. Understand that chromosomes contain DNA which carries genetic information in the form of genes. Describe a haploid nucleus as a nucleus containing a single set of chromosomes. Describe a diploid nucleus, two sets of chromosomes. State that in a diploid cell there is a pair of each type of chromosome, and in a human diploid cell there are 23 pairs. Describe mitosis as nuclear division giving rise to genetically identical cells in which the chromosome number is maintained. Role of mitosis: growth, repair, replacement, asexual reproduction. Then describe stem cells. Then state that meiosis is involved in the production of gametes. Meiosis is a reduction division. Chromosome number is halved from diploid to haploid. Understand that cancers form as a result of uncontrolled cell division. Now, how does DNA encode information? The genome contains information for the synthesis of functional molecules such as proteins and RNAs. The genetic material of most organisms is DNA. DNA is made up of a series of nucleotides. A nucleotide triplet that represents a particular amino acid of a protein is known as a codon. So we have A, T, A, T, G, C. Now this codes for one amino acid. This is called a codon. A series of codons is known as a gene. So a whole lot of these down here, a whole series of them will make a gene. Genes contain information about proteins or RNAs. The amino acid sequence of protein is determined by the codon sequence of a gene. So this will code for, say, amino acid number 5. Then another one will code for amino acid number 11, and another one will code so on and so forth. So the DNA codes for the amino acid sequence in a protein, and that will determine the entire shape of the protein. Now, what is genetic information? The summary is that genetic information is encoded in the sequence of the DNA double helix. To access this information, the DNA sequence must be copied. Why? Because the DNA must remain in the nucleus. It's just like a recipe book which I have, and you want a recipe, well, you must come and copy that recipe. I can't give you my entire book. The book must stay with me. So that is why the DNA or the chromosomes must stay in the nucleus, because we will repeatedly ask for the information how to make a certain protein. So transcribed of enzyme known as RNA polymerases, the resulting messenger RNA, mRNA, molecules carry the genetic information to the protein synthesizing machinery, where it is used to define the amino acid sequence and therefore the structure and function of proteins. So this specific polypeptide will result in this enzyme. Then a specific polypeptide will result in this protein or this enzyme. So every enzyme, why does it have a different active site? Is because its amino acid sequence is different and the DNA that codes for it is different. So this is how you have to understand. Like you can have a normal bread, you can have a banana bread, you can have a walnut bread. But the reason is because the recipe is different for a walnut bread and a banana bread and a normal bread. Now we come to another analogy. Genetic code, gene, genome. Then letters, word, sentence. So just a comparison to make you understand what is the genetic code. Those are letters, word is the gene, and sentence is the genome. So ATGC is the story which we're going to see in the DNA, and we'll talk about this in a further chapter, but we're not going to go into details as yet in this chapter. Now another very nice analogy. Analogy means a comparison. So DNA is like a cookbook. Genes are like recipes for proteins. Codons are like the words in the recipe. A, T, C, G are like the letters of the words. Amino acids are like the ingredients. RNA is like the cook. Ribosomes are like the ovens, and proteins are like the cookies. So just an analogy for you to compare what are we talking about. Gene, genetic code, genome, what is all this about? Now coming to chromosome numbers, there are two words that we use very frequently. One is say diploid and the other is haploid. Diploid means that every body cell of a human being has 23 pairs of chromosomes. So chromosomes are always in pairs. What do you have in pairs in your house? Your shoes are in pairs, socks are in pairs, gloves are in pairs. So diploid means the 2n, that is the double the number, diploid, double the number. So this now in this example that we are showing you here, the 2n number is equal to 6, or the diploid number is equal to 6. So what happens when we have a type of cell division which is called meiosis? In meiosis, what happens? The cell that is formed has only one of each pair. It has half the number of chromosomes. Haploid means half. So half the number of chromosomes doesn't mean it. In this you say 6, then here we have 3. But then this could also have been 3. But why is this wrong? Because this is a pair. In the haploid cell, in the cell which is haploid, which could be an ovum or a sperm, because these are gametes, it has to be one of each pair. Which one? Could be this one, could be this one, and this one, or could be any other one of each pair. So one of each pair is present in the haploid, and diploid is the double the number of chromosomes. Now I'm just giving you an example because some of you think that every organism has 46 chromosomes. No, every species has a different number of chromosomes, and there is no question of it if it's big or small, or if it's going to have more DNA or less DNA. It's not a question of that. Now, if you look at it, gorillas have 48 chromosomes, elephants have 56 chromosomes, donkeys have 62 chromosomes, a dog has 78, a guinea pig has 16, a pig has 38, a human being has 46 chromosomes, a cow has 60 chromosomes, butterflies have 380 chromosomes. So it's not a question of the size or anything which determines the DNA. So they can be different numbers. So chromosome numbers in different species: buffalo 60, cat 38, cattle 60, dog 78, donkey 62, goat 60, sheep 54. So chromosomes numbers are different for different species, and this is something which you need to be very clear about. Don't think every organism has 46 chromosomes. They can vary. They are very, very small numbers and very, very large numbers as well. Now, if you take out a white blood cell, take a blood sample and take out a white blood cell, and then we cut up the nuclear membrane and remove the chromosomes. Well, here's a diagram of what we get. And we've arranged them in pairs. And of course, we've used a dye to color them. Chromosomes don't have a color. So we've used a dye to color them. Now you can see they're in pairs. Now this is one pair. This is another pair. That's another pair, which is the same length and the same shape. So we have 22 pairs of chromosomes. And these 22 pairs of chromosomes code for the body characteristics. And in study in A levels, they are called autosomes. But this last pair, which is the 23rd pair, is actually sometimes it's a pair and sometimes it's not a pair. Pair I means is if they're both X, then it's a female. If it's a human cell that we are investigating, then because in some organism, please remember the male is XX. So in humans, this is a female. But if it is one larger, one longer or lengthier, and one smaller, then we call it XY. And this would be a male human being. So from a blood sample of a human being, without knowing the name of the person, we can find out the gender of that blood sample. We can see, okay, this blood is from a female. We can say this blood is from a male because they will be XY in a male and XX in a female. We've labeled it XX. We as biologists have labeled it XX. There's no X written on it or something like that. So if one is a little lengthier and one is a little shorter, then it's XY. But if both of them are the same length, then it's a female, XX. Now, one of your syllabus points is what is the role of mitosis? And what is mitosis? Let's talk about mitosis first. So what is the role of mitosis? The role of mitosis in growth, repair of damaged tissue. Please remember, not damaged cell. Do not say damaged cell. A damaged cell cannot be repaired. Please remember that. And that's an MCQ which came in one of your papers. So repair of damaged tissue, like for instance, you're using a knife to cut an apple and you cut your skin and the skin slowly repairs. So repair of damaged tissue. Your skin is cut, maybe a little bit of the underlying tissue is also cut. Like we do surgery, you do an appendectomy. So we cut your skin, we cut the muscle, and then slowly it heals and it repairs. So repair of damaged tissue is by mitosis. Replacement of dying cells. What does that mean? Red blood cells die every day. And new red blood cells have to be formed. Where have they to be formed? They have to be formed in the bone marrow. In the bone marrow, we have stem cells, and those stem cells divide. And then some of them still remain so that they can keep on dividing in the lifetime of that person. So, replacement of dying cells. And the last one is asexual reproduction. Asexual reproduction is like when you are planting sugarcane. So, you cut up the sugarcane and plant it in the soil, and it will start to grow. You take a twig. Sometimes there are some plants which we do by propagation. So you just cut a piece of stem and you plant it and it starts to grow again. So asexual reproduction. In mitosis, what has to happen? There is an original cell which has 46 chromosomes. And then this 46 chromosome, this cell divides and two cells are formed with 46 chromosomes in each. So the parent cell has now formed two daughter cells. Parent cell is finished. Why? Because we've divided that parent cell into two daughter cells. So one cell, another cell. In mitosis, we say the daughter cells which have been formed are genetically identical. Why genetically identical? The reason being is that all the chromosomes have to be copied. So first of all, the 46 chromosomes in one cell, that cell becomes 92 chromatids. And then it divides into two cells with 46 in each. Now, what are chromosomes made up of? Chromosomes are made of DNA. So, what is this process going to be called? This process is going to be called DNA replication. DNA replication, in which a replica is an English word meaning an exact copy, like we have Lady Diana's replica in the Wax Museum, Madame Tussauds in London. So, DNA replication, replica means an exact copy. So this 46 to 92, why? Is because the chromosomes are doubling. So if the chromosomes are doubling, then what is doubling? What are chromosomes made up of? DNA. So DNA replication has to take place, and the one cell becomes one cell with 92 chromatids, and then the cell divides into two cells, and then they have 46 in each. So this you have to understand is that the genetic information first needs to be copied, and then the cell has to divide so that half of the information, so that then now we have double information. So now we have a photocopy of the original paper or the original chromosome. So the 46 chromosomes were the original. Now we have a photocopy of them. So now they will go into two separate cells, and that is called mitosis. Now coming to the topic of meiosis, it's also called reduction division, and the name reduction tells you that what is reduced is the chromosome number. So in a human being, everywhere in the body, mitosis is taking place. But if this is a man, then in the testes, meiosis is taking place. If it's a man in the testes, meiosis is taking place. If it's a woman, then in the ovaries, meiosis is taking place. So in the rest of the body, in every cell of the rest of the body is mitosis. Mitosis is taking place. But only in the reproductive organs like the ovary and the testes, meiosis is going to take place. Okay, what happens in meiosis? Now, first, let's understand that. Now, if you look at meiosis, that is why meiosis is divided into stage 1 and stage 2. There is meiosis 1 and then there is meiosis 2. Now, first of all, as I told you earlier, this has to take place. DNA replication has to take place. So the 46 chromosomes become 92 chromatids. And then meiosis 1 takes place and two cells are formed with 46 in each. But in this situation also, please remember, it is still one of each pair. And then another two cells are formed, and now four cells have been formed with one of each pair. One of each pair. So this is what we have to understand, is that the 46 chromosomes become 92 chromatids, then two cells form, and then four cells form. The final result is four cells with half the number of chromosomes, with 23 chromosomes in each. And this process is taking place in the ovaries and in the testes. And that is why it is called reduction division, in which the chromosome number is halved. Now, why is it halved? The reason why it's halved is that the sperm is going to contain 23 chromosomes. That's the male gamete. The ovum is going to also contain 23 chromosomes. Now, when these fuse, one cell, the zygote, is formed with 46 chromosomes. So now this cell, which is called the zygote, is the beginning of every human being, every organism, every lizard, every frog, every insect, every fly, every mosquito. So this zygote is now going to divide by mitosis and form two cells. Then it is going to form four cells, and then it's going to form a ball of cells, and then it's going to be called an embryo, and then it's going to develop the human form, and it will develop into a fetus. And it remains a fetus till it's born, and when it's born, of course, it's called a baby. So zygote, then embryo, and then fetus. So what you have to understand is, why did this have to happen? Now, if this did not happen, whenever I ask you a question in the exam, I always say, when it says, what is the function of this some item, maybe what is the function of the diaphragm, and then you think, okay, if the diaphragm wasn't there, what would have happened? So similarly, let's look at it. If this reduction division did not take place, then what would happen? Now, as I said, if reduction division did not take place, so that means the sperm would have 46 and the ovum would have 46. Now, when the zygote would be formed, there will be 92 chromosomes in it. So, in the first generation, chromosome number was doubled. In the next generation, it would be 184. In the next generation, it will be 368. I know your mental maths is very poor. So 46 plus 46 would be 92. 92 plus 92 would be 184. 184 plus 184 would be 368. So every generation, the chromosome number would double. Now, why does it not double now? Why does it not double now is because every sperm and ovum has half the number of chromosomes. So when the zygote is formed, it is going to have 46 chromosomes, which is the normal for every cell of a human being. So this is what you have to understand, is why in sexual reproduction do we need to have reduction division? Why should the chromosome number be halved? So that at fertilization, the diploid number is restored. This is the English I want you to write. I keep on saying these days to all my students is that you must develop the correct English. So the diploid number is restored. The diploid number in humans is 46. In another organism, it could be 60. In another organism, it could be 80. So whatever the chromosome number is, that must be restored. The chromosome number should not double in each generation. If this had happened, then from Adam and Eve up till now, the chromosomes numbers would be in trillions, or I don't know, whatever figure that means, trillions or whatever comes after trillions. So at fertilization, the diploid number is restored. Now let's talk about stem cells. That is the topic which is in your new syllabus, stem cells. What are stem cells? You see, when the sperm and the ovum fuse, what is formed? One cell, the zygote. This is the zygote. Zygote divides into two, then into three, then sorry, two into four, four and eight, and it forms a blastocyst. And then it forms an embryo. Well, in the blastocyst, you can see there are stem cells which are derived from the inner cell mass. And these cells, which are stem cells, can develop into any organ of the body. They can develop into the bones, they can develop into the muscles, they can have blood cells, they can form the liver, they can form the neurons. So these are called totipotent stem cells. Now, this is a little higher level. I am just mentioning it, but you don't have to really know it. And then pluripotent are the ones you see from the totipotent. Some cells have become the placenta, but the rest of it, which are the embryonic cells, these are called pluripotent. Plural means they can develop into any organs of the body, the muscles, the blood cells, the liver. And multipotent will then be in the bone marrow, the cells which are going to develop into the blood cells, like the red blood cells and the white blood cells. What are stem cells? Stem cells are undifferentiated cells with the capacity to both differentiate and multiply into the 200 cell types that form a human being. Now, why is a lot of stem cell research being carried out? Why? Because we need organs. Somebody needs a kidney, somebody needs a liver, somebody needs a heart. What if we can use the stem cells of that human being to then form their organs, whichever they need? If we can form their kidneys, then we don't need to take a kidney from another person. We can do a kidney transplant of the same DNA. So that is why a lot of stem cell research is being done these days, so that we can then have organ replacement, so we can then replace organs which have failed, and then people would not have to go through the trouble of dialysis or something else which is going to compromise their life. The next topic is cancer. Now, what we have to understand that this is a healthy cell. Normal cell division takes place and healthy tissue is formed, right? But sometimes this healthy cell undergoes genetic changes and this becomes cancerous. It forms a cancer cell, and then the cancer starts to divide into two and then into four. Now, you know, cancer cells are immortal, they don't die. And ultimately, this cancerous cell division results in a malignant tumor. And then some cells of these can break off and enter the blood, and then they can be carried to some other part of the body. Like, for instance, this was in the stomach, and now they go to the liver, and then these cancer cells start to divide in the liver and start to grow. And that is what we call when we say that the tumor or the cancer has metastasized, and now it is found in the liver or maybe in two, three other organs of the body as well. And that is when we can't do much for this person because it has spread so rapidly from one organ to the other. So, what are cancers? Cancers are uncontrolled cell division. Cancers are uncontrolled cell division. So, uncontrolled mitosis. Mitosis must be done with a certain time frame. Like your skin cells divide every few weeks, your intestinal epithelium divides and is replaced. But if it's uncontrolled cell division, then it's going to result in a mass of cells which is called a tumor. But if this mass of cells does not spread, then it's a benign tumor. Benign tumors are not bad, they just occupy space. So we just need to remove them. But the ones which spread through the blood or the lymph, these are called malignant tumors, or they're called cancers in our layman's word term. So, what are cancers? They are just cells which have genetically changed and now they are going to result in uncontrolled cell division. Now, what causes them to mutate could be chemicals like tar and cigarette smoke, then some infections, viral infections, then exposure to UV light, radiation, and radioactive material. The normal cell undergoes a DNA change, results in a defective cell, cells multiply, cancer. Genetic changes and uncontrolled cell growth leading to tumor formation. Another diagram showing you this: normal cells divide, normal cell division eventually stops. But cells damage DNA, cancer cell division, and the cells keep on dividing and keep on dividing and keep on dividing. Now, what is asexual reproduction? Asexual reproduction, in which the main plant is there, and then the stem just goes off, and then a new plant starts to arise, and there's another plant starts to grow, and you can cut this here, and there'll be two separate plants. So these are different types of asexual runners, stolons, suckers, and offset. So they will just grow on and on and on, and there are no male and female gametes, and they will be genetically the same. They will all have the same genetic predisposition. There will be no variation. This is asexual reproduction. Now, asexual reproduction, plant propagation can be benefits quicker, assures characteristics passed to the offspring, less expensive. Drawbacks: can't be used with all plants, and there is no genetic diversity. needs only one parent. There's no fusion of egg or sperm and no mixing of genetic material. The offsprings are genetically identical to the parent. They're exact clones. Now, in asexual reproduction examples, most animals reproduce sexually: fly, human, snake, frog, all flowering plants and those that make seeds reproduce sexually: daffodil, grass, rose, oak tree. Asexual plants that make spores like mosses and ferns, fungi make spores, single cells which reproduce by binary fission or splitting into two amoeba and bacteria, budding like hydra, runners like grass, and fragmentation like planarians, and regeneration like starfish. So these are the examples of sexual and asexual reproduction. Another thing about the advantages: no need for a mate, takes less time and energy, reliable, few steps, produces large numbers of offspring very quickly. In stable environments with very little change, well-adapted organism can spread and colonize quickly, tends to require less parental care. Disadvantages: very little genetic variation in a population, harmful mutation in parent will be passed on to all offsprings. Entire population of genetically identical organisms can go extinct if there's a change in the environment. Like if they are resistant to drought and there's no rain for 2-3 years, then all will die. And this, of course, resulted in the famous famine in Ireland, in which there was a potato famine because all the potatoes were genetically the same. They were clones. So there was a fungal disease which destroyed all the crops, and the economy of the entire country was at stake, and it was a big problem. And that's when women started to do crochet and sell those crochet items in different cities of Europe so that they could earn something as a livelihood because the entire potato crop was wiped out in the potato famine in Ireland. Now, coming to the last topic, advantages and disadvantages of sexual reproduction. Advantages: yes, it's a variety. If you have one couple, a husband and wife, they have 4 sons, all of them are very different. All of them can be weird, all of them can be different. They're very, very different. So reduces the chances of offspring inheriting a genetic disease, leads to continuous evolution. Disadvantages: there's a chance of not finding a mate. Sexually transmitted disease can be easily contracted. There's no guarantee that the offspring will be born. So these are some of the advantages and disadvantages of sexual reproduction. Now this completes 16.1 and 16.2 of chapter 16. And we will continue the rest of it in the other videos. And please remember, these videos work for O level, GCSE, and IGCSE. So all those students who are studying either O level or GCSE or IGCSE, they can follow these videos, and the slight differences in syllabus, but not the major. 95% it's the same. So everybody, all those students taking these different exams, can follow these videos and be helped by them. And thank you once again. And thank you for watching, and thank you for subscribing, and wish you all the very best.